And speaking of great information, I think we're all very excited to hear David's update, his research update. He said he would do a song and dance for us. So he didn't say it would be good. He just said he would if I wanted him to. So, so David, are you ready to come up and give us our research update? All right.
Hello everyone. Hi. I'm animated, so I'm going to walk around a little bit. So hi, thank you so much for this opportunity, and thank you for the opportunity to actually represent the Alliance, or to help the Alliance in terms of being a part of your Scientific Advisory Council as well. So I'll start this off. I'm very aware that I speak quickly, and I'm very animated. I'm also aware that there are a lot of different languages in this audience. So when I speak about things, I'm going to try to start a little slowly. We're going to get into a little bit of detailed type things. If you miss some of the things or something's not quite clear, I'm going to bring it back together conceptually at the end in quite a simple way to try to explain sort of where we've come in research and why it's very exciting. So the first thing I did when I was asked to do this was I went through the about 2,000 papers that had come out in 2018 to see what could be true breakthroughs in ALS. And I think why I brought it back to a conceptual idea of where we've come in the last 6 years is because what we see in science maybe as a breakthrough isn't necessarily the thing you're going to see at a lay level from the media, for example, or something that goes out widely. But some of the things that have been discovered in 2018 are really prominent to the story that we're building. And so what I'm going to do is I'm going to bring us conceptually over a period of time, the last several years, and use mostly 2018 evidence. So you'll see a lot of papers flashed on the screen or manuscripts, and that'll be sort of the 2018 backup to say, you know, year over year, we are continuing to build on the story of ALS research. And in the last 6 years, I think it can truly be said that at a scientific level, the findings, the discoveries, the advancement has been pretty revolutionary in ALS. So why 6 years? Well, it's pretty arbitrary. Could be that I'm from Toronto and we call it the 6. The 416 is our area code. Could be that I'm looking back to Chicago 2012 as the 6 years ago. Their area code adds up from 31226, but it's neither of those. It's actually a selfish reason. And it's that 6 years ago I started in this role, coming from a laboratory bench, working on ALS. And the first meeting I went to in this role was in Chicago. And the idea is, if I sat with myself at that meeting and I told Dave of that time what we can do and what we have learned in the past 6 years, I think my mind would be blown. So I want to be able to share that with you. And so this is Jerry Seinfeld. He's a comedian in the United States. And so he had a really popular TV show, and he had a lot of jokes he did on stage from that show. And then he had a documentary where he kind of tore it all down, and he said, it's time to move on. I'm going to come up with new jokes. And it was rough. So he went up on stage and he bombed a lot. And what I'm going to do today is actually something completely different from what I've been talking about and building over the past 6 years, which is a very polished presentation. I gave part of it in Dublin a few years ago. And I've torn it down because I think the research has surpassed that presentation. It's come to a new side of things. And so I have to dream it all up again, and you're going to get the pilot of that. So I apologize if anyone has tomatoes, please have bad aim. So we use advanced technology here, and that advanced technology doesn't really exist, but fictionally it's kind of mind-melding. So Star Trek or this or whatever. And what I'm going to try to do over the course of this next half hour is to get you to see inside my mind and why, from a more holistic view of not just understanding ALS, not just clinical trials, but how have we built a foundation to really truly having hope, and why I'm so excited about where we are in ALS research. So I'm going to avoid all the crazy things that are in my brain, and we're going to focus just on the ALS or the MND portions and how I feel we're really surrounding it from a lot of different levels now in 2018. So the first step is, if we think about those true breakthrough periods in ALS, my mind kind of goes to a series of buckets, and they kind of go back in 6-year periods to some extent. So we have the BS phase, or before SOD1 phase, where this is sort of how I see things. There's a lot of shots in the dark in terms of trying to understand what's happening with ALS. Then from 93, when we discovered the first genetic cause of ALS, there was this palpable feeling that we may be able to treat this disease or potentially cure it in the next several years. It then wasn't so simple because we really needed more tools to work with in the laboratory. And so this BT phase was before we discovered sort of the next major player, the next major tool in research called TDP-43. Then there was a phase where we started to discover more of these genetic causes of the disease, and it was a hopeful time leading to what I would say in the last 6 years, which is sort of a time of confidence, where I notice a palpable difference when I go to an ALS meeting in terms of the feeling that we're getting somewhere. Now, the researchers in the audience that are here would probably say, we have a long way to go. It is a very complex disease, and I'm not trying to sell anything different than that. But I really, truly believe that we are on the right path. And at the very end, I'm going to wrap it up with a diagram that should show you, inside my brain at least, where I think we've come just in the past 6 years. So one of the analogies I'm throwing out is the puzzle analogy. And it's an important analogy, but I think we've surpassed that. And the idea really was that ALS is like a puzzle. And for many years, we really only had one of those puzzle pieces. And it's a very complex puzzle to try to understand with one piece. And over the past several years, and between 2006 and up till now, we've been discovering more of those puzzle pieces, which are tools to work with in the laboratory, essentially mutations in genes that can cause a form of ALS or cases of ALS that we can use to try to understand the global aspect of the disease. And it would become clearer the more of these that we discover. Thing is, I think we're getting past the phase of gene discovery, and now we're getting into the idea of how do we put those pieces together, and how do we look from above at the picture that's being formed. So some slides that I have used in the past that I think I'm going to keep, which is to explain to an audience, well, how do we use these tools in the lab to try to find therapeutic targets that can work for the disease? Now, these are the four most prominent genetic causes of ALS, or at least the proteins that those genes create. And the four most prominently studied proteins in ALS. And it's not important to know what they all do. We're not going to get into that. But basically, through some mechanism, when you have a mutation in SOD1 or FUS or TDP-43, some biological activity is happening that is causing motor neurons to degenerate. And when you look at these together and when we learn more about them, we're going to start to see connection points that are potential therapeutic targets for the disease. But the cool part is that now that we've discovered many more of these puzzle pieces, we can learn about each one of those, and that's going to start to fill it in and show us whole systems and how they connect into a much greater overview of what's happening in ALS. And I think in the last few years, this is where the picture's starting to become clearer and clearer as to where we might be able to therapeutically target the disease in a more effective way in the future. And just to give you an idea, that one puzzle piece idea, when we were looking for many years on just SOD1, and that's the only tool we had, it's all of this other stuff existed, but we couldn't see it. The people out there living with ALS with these types of mutations or these abnormalities going on in their motor neurons, it was all there, but we couldn't simply see it, so we weren't able to solve that puzzle. Additionally, it's entirely possible that ALS is a disease where many of these are separate diseases that all result in the same endpoint, which is motor neuron degeneration leading to ALS. And I would say even 2 years ago, I felt like, boy, that's a very daunting idea. But I think I really believe now that this is still something we're going to be able to do, even if this is many different diseases that funnel into the same endpoint, or if it's 3 or 4 diseases. I think it's entirely possible. And we're already doing that at some level because there are now clinical trials happening that are targeting specific forms of familial ALS. And it could be that there's maybe just 3 diseases or 4 diseases, or it could be that a therapy could work for all of them. And we're learning that, and we are getting closer to starting to suss those things out. The last slide from the Dublin presentation that I'm giving here was that in 2016, I was pretty excited because when I look into the literature, I started to see all the connections that we've been finding between these different tools that we're working with. And that was pretty exciting. But I think we've gone beyond that. And I think now we're at a point where we can start to see that point at the end, which I was talking about, where the whole systems are coming together and we're starting to see connection points. So I just reiterate that when we talk about breakthrough, I would say in ALS, maybe there are 3 or 4 big breakthroughs that we've had in discoveries over the years. But in terms of what I truly believe are breakthroughs, I think every one of these large findings we find that maybe don't come out to all of you are breakthroughs for us, because they are helping us to build a story and to see the puzzle better. So we're going to do this quickly from a before and after model, thinking about Chicago 2012 to Glasgow 2018. I think people are familiar with the before and after model. It's quite frequently used for weight loss and things like this. So we're going to go with that for this particular thing. And I'm going to give you my 6 arms of the foundation that we're building. The first one, well, understanding ALS. And I must say that these are just examples off my head. There are more things, and you cannot know everything, so I'm happy to have people come up after and say, you know, I think this should have been included. As I said, this is a developing presentation. So the first one really stems off what I've already been talking about, which is that in 2012, we really had information on 3 of these genetic causes of ALS, and now we're starting to get more information that's coming from many of the other tools, which are helping us to see a clearer picture. Now, bear with me. The first arm of this is the more complex one. It's the one I'm going to focus on a little longer. The other ones are going to be much shorter. So if you're getting through the first one and you're getting tired, it will go faster after that. So just to give you an example of a 2018 discovery that comes from one of these new genetic causes that we've discovered in the past few years. In 2010, there was a gene mutation that was discovered for ALS in a protein called optineurin. Now, one of the things we needed to do was to discover what are the roles of optineurin. And it was discovered that one of the things it can do is inhibit something called RIPK1. And RIPK1 is bad for motor neurons. Let's just say it causes motor neurons to die. Now, if you have a mutation in optineurin, it actually causes it to lose its normal protein function. So in this case, one of the normal protein functions was it wasn't able to inhibit RIPK1. So they produced a mouse model of this, which had reduced optineurin. And in fact, what happened was RIPK1 was activated, and you had motor neuron degeneration in those mice. Now, move along to 2015, and we discovered another new genetic cause of ALS called TBK1. Well, when we looked into what was known about TBK1, it was actually something that had a lot of biological activity connected to optineurin. So in 2018, there was a desire to say, well, what happens in a mouse that you have reduced amount of TBK1 or a mutation in TBK1? And it turns out, we found out that in fact, TBK1 also inhibits RIPK1, which we know is bad for motor neurons. So when they made a mouse that had reduced TBK1 activity, in fact, the mouse could not survive. So this meant that TBK1 had some effect in mice that was important to its development. That's fine. But when they reduced the levels of TBK1 to allow the mouse to be born, but then see what happens, it turns out that it did get a motor neuron-like degeneration. But there's an extra piece that came out of this discovery in 2018. And that was what they found is that when TBK1, which we know inhibits RIPK1, which is bad for motor neurons, when it is reduced in its amount or its ability to do what it does, there is a compensatory or compensating mechanism by something called TAC1. But what's interesting is that TAC1 actually reduces in its levels as we age. And so what they found is that when you have a reduction in this TAC1, which can mimic aging in a sense, you ended up having motor neuron degeneration in these mice that have the TBK1 mutation. So the cool thing is that this is the first time that we've been able to actually connect aging as a risk factor with one of those genetic tools that we're working with in the laboratory. And where does this sort of make any sense in terms of what we want to do with this? Well, it says that perhaps we should definitely be trying RIPK1 inhibitors in terms of moving this towards whether it has any therapeutic effect, first in models of ALS, and then potentially, if that works, maybe moving that to the clinical space. And why you may not have heard of that is that it's obviously very important that when we have these types of scientific findings and a potential therapeutic target, that we're not telling the rest of the world something too early, because we don't know if this will actually work until we try it in models in the laboratory. But I think it's very interesting, and it tells us that we can start to make some connections between some of these tools. And actually, another paper that came out in 2018, so this is a 2018 paper in Cell here, and another one that came out confirmed that TBK1 has an effect on this bad thing, RIPK1, which is always good in science when you can have replicated data to show that it's happening. So that's the last biological mechanism one that I'm going to show, but it's just to show you these are the types of studies that are being done. So it's sort of a must-try next to be able to see if these RIPK inhibitors might be important in ALS. Now, the second one that I came up with was that before, in 2012, RNA biology was something that was still in its infancy. And I think today our knowledge on this at every level has massively advanced. And just a quick bit of evidence on that. If we look back to the Chicago conference, there was actually a few talks that said we need to make an argument for why RNA biology is important in ALS. And what we found over the years is that a number of those genetic causes of ALS actually encode proteins that are what are called RNA binding proteins. And so over the years, we've started to realize these are likely very important to at least a portion of ALS cases. Now, in the past several years, it hasn't been that we've just understood these specific RNA binding proteins that are important in ALS, but we've actually learned a lot about RNA biology itself. So this is like a diagram from a high school textbook, and these are things that have been discovered that you would never even find in those old textbooks. The first one is called stress granules. So what we know is that these RNA binding proteins, they actually can bind to RNA, which is an important molecule in our cells. And actually, when there's stress in the cell, they huddle together, just like penguins do in the Antarctic. They huddle together until the stress is gone, and then they need to sort of break open again so that they can do their normal important functions in the cell. Unfortunately, what we learn in ALS is that the dynamics of the creation and breaking apart of these stress granules is not happening properly. So they're kind of stuck often in these sort of huddles, and then how are you going to be able to do proper things for your penguin colony, in this case, in the cell? And this is what they look like in cells. And beyond that, we've even further learned about a whole new bit of cell biology that ALS has been the forefront of the understanding of. And that is that these RNA binding proteins and RNA actually are like liquid droplets. Oh, that one got stuck behind there, but it's actually like oil and vinegar or like a lava lamp that's happening in there. And it's a whole sort of layer of biology that we're just learning about that's going to help us to understand what might be happening in ALS and how we might therapeutically target it. And even this year, a 2018 study used a new advancement in technology to be able to understand what is happening in these stress granules and what are the pieces of those stress granules. And those are things that we can eventually start to target for therapy. So I know that's very technical stuff, but ultimately what it's saying is that this is an important piece of the biology that we have advanced a lot on in the past 6 years. The third one I'm not even going to read because then it just gets a little bit too much. But what this is really to represent that thing I was talking about bringing systems together. And I promise you this is one of the last one of these sort of biological mechanism slides. But bear with me. Hopefully you can see some of the connections here that we've been seeing in 2018. Okay, in ALS, we know that in 97 to 98% of cases of the disease, something called TDP43 is supposed to be found in the nucleus. And my laser pointer is not working on the screen, but it's that central area on the left. And in ALS, it's actually found outside of the nucleus in what's called the cytoplasm in these clumps. And TDP43 is what's one of those RNA binding proteins I talked about. Now, we could say that perhaps it's not able to do its normal function in the nucleus, and maybe it's having some extra toxic function in the cytoplasm, and it's probably both at some level. So what we know is that when the TDP43 or other RNA binding proteins like FUS leave the nucleus, they can also form stress granules, like I talked about, the penguins, and also this liquid phase separation. Well, in 2018, a number of papers came out to show that when they're doing this, they're actually sequestering factors that are important for keeping things in their proper place between the nucleus and the cytoplasm. So now we're starting to make those connections to say, so something's wrong with the biology that they're trapping important factors that are getting them to the right places of where they need to go. And this actually then correlates with something we found a few years ago, which is that the most prominent cause of ALS, the most prominent genetic cause of ALS, C9orf72, actually causes defects in nucleocytoplasmic transport. So now we're able, and furthermore, I apologize there, in 2018, Dr. Leonard Petrocelli's group actually found that this C9orf72 also plays a role in the dynamics of these stress granules. So it's kind of hard to see, but what you can see is that there's a connection between all of these different levels of biology that we're starting to make from the discoveries of 2018 and of the last few years. Furthermore, this C9orf72, when there's a mutation in it, it's not only becoming toxic on top of it, it's not gaining an extra toxic function, but there's some evidence that it may also be losing its normal function. And what's interesting about that, and I won't get into the details of that, is that loss of normal function connects in some ways to other tools that we're using in ALS. And furthermore, we're even learning, here's a paper just from a few weeks ago in a high-impact journal, which is really saying that sometimes in some cell types, this movement out of the nucleus and into the cytoplasm is beneficial. So all this to say that from our standpoint as scientists, when we look at this, we're starting to see connections between those big systems that aren't just one gene or another, but actually how can we put the puzzle pieces together. And we're also looking into other areas of the world. So what are prominent gene mutations in China, for example, or in other countries in Asia that might tell us these are the next ones we need to focus on, because clearly they're going to be important for us to understand how they affect motor neuron degeneration and ALS. And ultimately to say from this that we're seeing those connection points come together. And I'm trying to put together a nice diagram of this. Like I said, this is a work in progress, and I will be able to come up with, hopefully, invite me back again, and I will have a much more easy to understand diagram which shows these connections. The next one is the neighboring cells around the motor neurons called glial cells, and I think we've really evolved in our way that we're going to target these for therapy. So in ALS and in our central nervous systems, you have cells that are surrounding the motor neurons that we've known for many years are very important to the disease because they actually become reactive and can cause a neighborhood around the motor neurons that is not great for their survival when the disease happens. And I would say that for the first several years, we sort of did a lot of blanket therapies that just said, well, let's tamp down or reduce this inflammatory effects that are happening. But I'd say in 2018, one of the things that we're starting to see is people targeting this more efficiently and saying what's actually going wrong in those other cell types. For example, this whole mechanism is happening in one of the cells outside of the motor neurons. And as we learn about those things, we're going to be able to target the glial cells or the neighboring cells more effectively in clinical trials. An example, ALS TDI has a drug that a therapy that they're moving forward to the clinic that is looking at a very specific activated mechanism or specific mechanism in these supportive cell types. And we have to think that they're going to be very important potentially in the next few years in terms of therapeutics, because if we can't diagnose the disease earlier yet, it will be very important to provide a nurturing environment for the surviving motor neurons to slow progression of the disease by targeting each of these different cell types in a more surgical and a detailed fashion than we have in the past. And finally, I think one of the biggest things that if I were to talk to my 2012 self and say, we've come a long way on, is if I asked myself then, what are the major hurdles that we have never tackled before in ALS? I would say looking at sporadic ALS in the laboratory and examining human ALS and why is human ALS different from one person to the other was a huge untackled hurdle and something very, very daunting to think about. And I would like to say that now we're actually making inroads on trying to do that. Things like Project Mine, Answer ALS, different precision medicine programs are starting to look at these big answers. I mean, I don't have to tell any of you that there are people who progress quickly, there are people who progress slowly, some have cognitive impairment, some have different types of cognitive impairment. different places of onset, and we need to learn this not only to understand the disease, but also to put clinical trials together in a way that's effective. And again, in Milan, now 5 years ago, there was a big call at the plenary session to say we need to start focusing on this heterogeneous idea for ALS. Last year, it was doubled down by Dr. Jeffrey Rosenfeld about the same thing. And I think we're really getting to a point where we're starting to be able to look at ways to tackle that, and that's pretty amazing because I think that we're going to learn an awful lot. So the second one, so that's the slog. If you got through that, it's going to be easier from here on out, I think. The second one is collaboration, and that comes from what I was just talking about with things like Project Mine, Answer ALS, etc. And I'm going to go through these ones not actually individually as I'd intended, but really just going to talk about them very briefly in an overview here. So for example, in 2012, Project Mine was still not something that had been started, and here we are 6 years later, and it's getting closer and closer to its goal. It may seem like it's been around for a while for many of us, but in fact, 6 years is pretty fast to do a study like that when you're talking about ALS, which is not a disease that receives an awful lot of funding compared to other ones that are more prevalent. Furthermore, 6 years ago, I would say that there were a lot of multi-investigator studies, so individuals who are collaborating with each other, whereas now we're seeing multi-investigator platforms, big national platforms that are tackling big subjects in ALS that we weren't able to look at in the past. And those are resulting in a true a big data era for ALS, where we're starting to get amounts of data about ALS that a human can't actually comprehend, and we're going to need to look at new ways to be able to analyze that, and I'll get to that in just a brief moment as well. And furthermore, we're looking more into this idea of open science. So now it's about data sharing, and I think one of the key pieces in ALS is how we're going to be able to leverage each other by sharing our data, pooling it together, and getting bigger values so that we can actually start to understand things with significance. And the next one as well is that if you look back to 2012 or previous years, I would have said that in ALS, it was great if we had a few papers that were in high-impact journals, and now it seems to be a couple times a month you get something that's in a very high-impact journal, and that's a testament to a couple of things. One, that we've made big inroads in our understanding of the disease, and that deserves to be in high-impact journals. And second, we're starting to see that collaborative effort where individuals are saying, let's answer a big question and let's get that person from this place and that other person who's an expert in that and bringing them together to really answer and tackle big questions. And then clinical trials is another big one because I think 6 years ago, you ended up having a lot of clinical trials that were still country-based, and we're getting closer and closer to this idea of multinational clinical trials. It's happening much more often now, and I think that this, again, shows how collaboration is important. Visually, I think many people know of Project Mine. This is a great example of how countries can come together and leverage finances and resources to do something really big on an epic scale, something we need to do to make big answers. Answer ALS is going to produce an amazing amount of data about ALS and human ALS. As will Project Ambrosia in the UK, Pulse in France. Neurobank in the United States is trying to bring people together, even here in Glasgow, to be able to say, how can we take these platforms and pool the data together? How can we figure out how to bring it together so that we can learn from each other's different platforms? A platform from the ALS Association, who is then connected with ourselves at ALS Canada and the MND Association to look at a specific tool we use in the laboratory that has caused a lot of delays in science over the years, and how do we do that better? How do we make that more efficient with this ALS reproducible antibody platform? A PROACT database is a database of all of the clinical trials that have happened in ALS so people can mine this to learn about the disease in a better way. And I should have put also PACTELs on there, but PACTELs, NEALS, NCALs, they are leading the way in terms of collaborative efforts to drive the field forward. And that leads us to the third arm, which is clinical trials. If we look back to 2012, we're in a situation where Biogen had a huge imprint on the Chicago conference. They were just finishing up a clinical trial in phase 3 of something called dexpramipexole, which had positive phase 2 clinical trial data and sadly failed or had to be announced as a failure for the disease in general in early January of that year, of 2013. And now we're in a situation where we have something that is another drug for ALS, and it's starting to say that there's dents in the armor in terms of our ability to potentially slow progression of ALS. And I think that there are things in the pipeline now from the learnings that we've had from this Biogen trial that are leading people to get closer and closer to being able to have more therapies for the disease. I should say, probably immature is maybe not the best word, and I think people who have been working on this, I definitely don't want to offend anyone because there are amazing people who have been working on trial design, but I'd say one of the biggest advancements over the past 6 years is the extreme effort that's been put forward in doing better trial design in ALS. And this is increasing the efficiency, the effectiveness of trials, new outcome measures, new way of running trials. And, you know, I can say this up here, but I think if you go to the plenary session at the symposium, you'll see Dr. Hiroshi Mitsumoto, who will talk about the clinical trials guidelines, which I think many of you leveraged clinicians from your countries to come together and build an international effort to say, how should clinical trials be run effectively in ALS? And I think that's a huge learning from the Biogen situation to say that we need to do these in a different way, and it has come so far in the past 6 years that I think it's truly remarkable. Things like ASOs, antisense oligonucleotides, were sort of just in the early phases back in 2012. Now, 6 years later, there are full steam ahead with therapies on SOD1, C9orf72, and soon to be even more targets that might even be related to genetic aspects of sporadic ALS. Brainstorm, for example, was something that was sort of a company in Israel that had not a huge knowledge about it globally. There were some media releases at the time, and here in 2019, we're getting closer to the time when we're going to find out from the first time a phase 3 clinical trial of a stem cell therapy as to whether or not there's value in that moving forward. And in 2012, there were a couple of high-profile trials, and I would say in 2018, well, if we have a quick look at that, it's pretty amazing. And if I'm missing anything in here, I apologize for that, but Ionis and Biogen, Ionis Pharmaceuticals, are moving forward both SOD1 and C9orf72 antisense oligonucleotides. Like I said, what is this? These are creations that can actually reduce the level of a potential toxic gene or a toxic product, and then delivered intrathecally into the spinal fluid to be able to see if this will work. And the SOD1 trial is probably moving into phase 3 soon, finishing up the results of a phase 1, and they are currently recruiting for a C9orf72 phase 1 clinical trial. What's amazing about that, this is only 7 years after the discovery of that gene, and all of the way now, we actually have, in 2018, clinical trials directly targeting this. Wave Life Sciences are working on antisense oligonucleotides for C9orf72. Pfizer and Sangamo have shown interest in targeting C9orf72, the most common genetic cause of ALS. Avexis Therapeutics are looking at a viral mechanism that has already shown promise in clinical trials for spinal muscular atrophy. I should also say that the antisense oligonucleotides are the first approved therapy for spinal muscular atrophy in children with Spinraza. And Avexis is using this in terms of a virus that you can infuse IV one time only, and it has long-lasting effects. And they're targeting SOD1 to start, moving into clinical trials. Brainstorm, as he said, is the first time we're going to see a phase 3 clinical trial results on a stem cell therapy. Basically, what they're doing is removing mesenchymal stem cells from bone marrow of an individual and then culturing these in the laboratory and treating them with a compound called Neurone, which is boosting the growth factors of these cells, and then returning those into the cerebrospinal fluid to see if they can slow down progression of motor neuron degeneration. Furthermore, just a few months ago, there was a publication in South Korea by a company called CoreStem and a group that are looking at doing a stem cell type therapy as well and showing promising results from a phase 2 clinical trial that they've run in South Korea. And now they're looking at whether or not they can move this into a larger trial somewhere else outside of South Korea. So also promising in the potential for stem cells moving forward in organized and rigorous clinical trials. Cytokinetics, Orion Pharma are looking at muscle. As we know, tirasemtiv. We now into the next generation of that with reldesemtiv from Cytokinetics, and levosimendan from Orion is something focused on how the muscle responds to motor neuron signals. Mallinckrodt is in the field working with a synthetic hormone called Acthar gel, which is trying to tamp down on the inflammatory and immune responses. Speaking of that, AB Science has something called masitinib that should be moving to phase 3 clinical trial now soon, and they had promising results in a phase 2 clinical trial in slowing down progression of disease. Analyxis Therapeutics, which is the first time that ALS TDI has moved something from their in-house work that they've been doing for many years through the translational spectrum, and this is the thing that they've seen should work or has a good chance at working moving forward into the clinic using something called an anti-CD40 ligand antibody. Procypra Therapeutics in Australia is working with an interesting molecule called copper ATSM. I could talk about that at length, but I won't go on with that. Genentech is working on something called DLK1 inhibitors in a phase 1 clinical trial moving forward. GSK is still interested in ALS. They had a clinical trial a few years ago that didn't do well called ozanezumab, but they still have interest in ALS and I believe are moving some things forward. And we also certainly have Mitsubishi Tanabe Pharma, who are not only optimizing the work that they've got with Edaravone in terms of delivery mechanisms and other ways in which this can get to people, but also continuing to look at a pipeline on how they can continue in the ALS space. And looking back to 2012 and looking at 2018, I think it's pretty amazing at the interest in coming into this space by pharmaceutical companies in terms of looking at therapeutic options for ALS. Biomarkers is something that I'm not talking about here, but I believe if I were to give this talk in a few years, it would be the 7th arm, because really understanding biologically how we can measure prognosis of disease, how it progresses, and potentially diagnosing the disease earlier. This was something I saw, which is a prospective study in the UK, which will help us to potentially someday find diagnostic biomarkers for different diseases. This is going to be the way of the future, and I think biomarkers is something I could talk about as an entire presentation unto itself. And even just recently, there have been a huge drive by NEALS in the United States to start doing platform clinical trials, which is a whole new idea where you have one placebo arm and several different therapeutics measured against each other, but also reducing the number of people in ALS who are on placebo. Which is pretty amazing, extremely difficult to do, but I think they're trying to move that forward because it's necessary. The last few here, I'm going to do it very quickly because I am not an expert on these things, but I think one of the arms should be quality of life. You know, obviously engaging people who are living with ALS is super key to us getting to a world without the disease. Not only are they the key donors or the key advocates, but they're the ones that are hopefully going to benefit from these therapies. And as we find therapies that we can slow down progression of disease, you need to meet that with a rigorous amount of quality of life for these individuals. And I think, you know, these first two are really saying that there's much more emphasis in the field of research to say we need to put forward a better consensus on objective measures for symptomatic management of ALS. Guidelines are being improved. There are more rigorous clinical trials on symptom management than ever before. And I think there's a much bigger emphasis on this. Telemedicine back in 6 years ago was sort of an idea, and now it's starting to actually be used in certain clinics. Also teleresearch, so research from the home so that people with ALS don't need to continually come into the clinic. We're trying to advance those things in the field. I think even 6 years ago, there was still some skepticism between how much linkage was between ALS and cognitive impairment and frontotemporal dementia as a spectrum. And I think we're getting closer and closer to that being a widely accepted thing that needs to be tackled, and we need to understand it. In fact, right here in Scotland, there was development of a measure called the ECAS, or Edinburgh Cognitive and Behavioral ALS Screen, which is a really great test to be able to sensitively measure whether or not someone with ALS has some level of cognitive impairment that they can then go on to further assessment. And this actually, in terms of quality of life, you can imagine as a caregiver how valuable it would be to know whether or not your loved one potentially has something that is maybe changing their behavior or their cognitive impairment that isn't controllable. This is something that's actually part of the disease. And brain-computer interface is something that I think was talked about and conceptual 6 years ago, and we're getting close to it being a near-market reality. And I'll show you one quick example moving forward. And these are just some 2018 papers that support that, looking at quality of life, a better belief that we should be able to do better. I'm not a physician, so I shouldn't say we. I'm just a huge fan of everyone that's working on this disease and really putting so much effort in. But ultimately, as a field, people are doing these things, and they're thinking about this in a much different way. Furthermore, research is being done, as I said, at home, and you're thinking about TDI with their precision medicine program. Jeremy Scheffner and Stewart Rutcove have an ALS at home program. Richard Bedlack had a program where people self-report for their clinical trial in terms of how their functional decline is happening. Apps for at-home use. These are the way of the future so that people don't necessarily always have to come to clinic for clinical trials to move forward. The next one I actually would love to learn from a lot of you, and I think visibility is extremely important because how are we going to get the funding to be able to have a world without ALS, without better visibility? And I bolded the first one because I think we all understand that one big turning point in terms of visibility for ALS was the Ice Bucket Challenge, which did sweep the globe. But there are a number of other things that I think I could learn from all of you, which would help me to understand how visibility is being increased in each of your countries, and then how do we bring that together globally to increase this foundation that I'm talking about building. So again, I think there's a lot of work to be done on visibility because as we can increase that in the future, it's only going to help the other arms of this particular avenue. The last one is technology. And this is the one that I've chosen not to pick 5 individual ones, but just to show that technology is overarching all of these. And to me, as much as I am enthusiastic about what we've understood about ALS, as much as I'm enthusiastic about how far clinical trial development has come in the past 5 years, technology has really come a very long way. And let me just give you a couple cool examples of this, ones that I get excited about. So have you ever heard of Ibm Watson? You may have heard of this study. It was actually a supercomputer by IBM that takes a bunch of information and collates and looks for connection points between the information. So it beat some grand champions on the game show Jeopardy several years ago. And then they said, well, maybe we shouldn't just win at game shows, but we should develop a way to allow these computers to help us with health indications. And one of the people that they first talked to in the neurology side of things or in neuroscience was Dr. Bob Bowser, who's actually a ALS researcher. And in Dublin, 2 years ago, he first sort of presented this information that was now published in 2018, where he did a study to see whether or not IBM Watson could start to help with understanding ALS. Now, I'd said to you before that I just realized that I haven't been using the microphone very effectively. I apologize about that. Hopefully you can all hear me. I had said that a number of the genetic causes of ALS are RNA binding proteins. And he had known that by 2015, maybe 11 or 12 of these that were connected to ALS were RNA binding proteins. But there were actually almost 1,500 of these in biology. So perhaps other ones were important in ALS. And he wanted to see if IBM Watson can help us to find out some of these. Well, the first thing he needed to do was to test it and see, well, can it work at all? So what he ended up doing was feeding it all the information on these RNA binding proteins in ALS leading up to 2013. And this was in 2015, mind you. And so by feeding it all that information, he wanted to see if Watson could then spit out what was discovered in 2014 and 2015 as RNA binding proteins that are important in ALS. And sure enough, it actually predicted those discoveries that we had already made from information that was given only up to a certain point. So the next step was, well, let's give it all the information we know about RNA binding proteins up to that particular point, and it came up with some new ideas that he's currently validating. Now, that's an early study, but what it ultimately says is, can we start to put together a lot of the data that we have on ALS and say, are there connections that our human eyes simply can't see that machine learning and AI advancements are going to start to find correlations or potential therapeutic targets that are just simply in this grand scale that our human brains can't quite comprehend. One other really cool study like that actually comes from the idea of Google Deepmind. So I don't know if anyone's seen the documentary called AlphaGo, where they had a deep learning, a DeepMind actually played a grand champion of a very complex game called Go. I've been given the time signal, so I'm going to be very quick here. And ultimately, they decided they also wanted to go into biology. And they did a really cool study in 2018 that was published in 2018, which involved seeing how well it could work with microscopy. So this is a series of cells. We look at cells in the laboratory, and this is what it looks like under a light microscope. Can't see much as a scientist. When I look at that, I see a few things, but I can't really tell where anything is in there. In science, we can actually label individual proteins that allow us to see the structures and fluoresce under a microscope with fluorescence. However, we can only limit each one of those cells to be able to study a couple different proteins at a time. So what they wanted to do was to say, if they looked at a whole bunch of these labels and then looked at the light picture for each one of those, could they start to say that that DeepMind would actually be able to learn what you can see in that light image that tells us where everything is? And the idea was, and it came out pretty strongly on this, was if you could now show it just one light image, it would be able to tell you where maybe 10 or 20 or 30 different proteins are found just by simply having learned the correlations between something our brains can't see and what the computers can actually see in the image. And the cool thing here is that the person they engaged with on this is Dr. Steve Finkbeiner, who's also an ALS researcher. So this shows that ALS is at the forefront of this AI and machine learning world. Last slide, I promise, before I get to the last little concept, and then I'm done. It'll be like 2 minutes. CRISPR gene editing is something that's technology. Again, in 2012, I would not have even been able to fathom that it would be as commonplace as now. We can do quite easily in cells or even in animals. We can target a particular piece of DNA and we can just change that piece of DNA. We can add a mutation. We can remove a mutation. We can remove a piece of DNA. We can add a piece of DNA. All kinds of things we couldn't have fathomed being able to do 6 years ago. iPS stem cells. Now we take blood from an individual living with ALS and we can turn this into any cell type we want. And in 2018, there was an advancement on being able to take sporadic ALS samples and to be able to see some of the disease aspects coming out in these cells, which will help us to possibly, in the laboratory, learn about sporadic ALS. And we're even able to move these into small organ structures. Organoids and mini brains are things that we can do now that may have really good application in the future. Detection limits, so we can do things in single cells that 5 years ago we could only do in whole systems. Microscopy advancements, all of these are things in 2018 that are moving forward. Synthetic biology, the ability to actually create life in a small cellular organism is something that eventually will become reality in terms of, or potentially reality, and we don't know yet how that could be applied to ALS, but I think that's definitely something we can consider moving forward. And even cooler and even crazier is that we're actually on the cusp of potentially a huge jump in computing power. So after I've told you all that stuff, we're at a place where Google would say we're getting near what's called quantum supremacy. And we have conventional computers that use these ones and zeros for many years, and we're now onto this whole new series of computers called quantum computers that use something called qubits. And it's believed that they are soon going to surpass conventional computing mechanisms. And as that advances, imagine when we start to apply that to diseases like ALS and how we'll be able to actually learn about what's going on. Last, BrainGate, and Dr. Lee Hochberg actually gave me this a video to show today. This is actually brain-computer interface in terms of somebody who is completely locked in with ALS, who's imagining using a mouse to try and click on things. And this is amazing computing power and how far we've come. If you look on the left, oh, it's actually not, oh, it's going now. So on the left is the first 20 seconds of the computer trying to learn how an individual is is imagining clicking on that red dot, and you can see it's very slow. 3 minutes later, you can see how quick the accuracy is in terms of that computer having learned that individual's neural pathways and the ability to be able to click on things when they can't move at all. Pretty amazing things are happening in 2018. So conceptually, and I know I've been all over time, but this is the last bit, and this is where it all comes together, is that if we look at these 6 arms and we kind of consider where we've come from, back in 1938, don't worry, I'm not going year by year. If we look at 1938, before Lou Gehrig was diagnosed, a lot of these didn't exist. It was kind of a niche neurological disease. By 1939, there was some visibility around this, and I think there was an idea that we need to figure out how to therapeutically go after this. Very, very rudimentary clinical trials with vitamins happening, but at least the idea of trying to treat it. Moving ahead to, say, 1960, we first had a little bit of understanding about ALS because clinicians started to see that there was an actual hereditary potential for the disease, and it could be familial in some forms, which led to modern-day belief that, or modern-day ability to look at genetic causes of the disease as tools to understand it. Technology increased because you started to have power wheelchairs, which is obviously something that could help with quality of life at some level. Moving to 1980, we started to have ALS specialists, people like Forbes Norris. In Canada, we had Arthur Hudson and a number of others who started to see themselves as ALS researchers. And they started to come together and talk about things like multidisciplinary care. And this is obviously still the model today for the best way to be able to care for people with ALS. By 1994, we had, in 93, discovered the first genetic cause of ALS because of an increase in technology, the ability to actually identify these genes that cause the disease. So we had an increase of our understanding of the disease for the first time in a long time, and technology had increased. We also had increased in collaboration because people started to come in to try to work on this disease, that we had something to finally work with in the lab. Moving to 2000, I would say the understanding hadn't moved that far. We certainly learned a lot about SOD1, but looking at modern day, how far have we really come? Because we only had that one puzzle piece. But clinical trials started to develop a stronger aspect. You started to get collaboration between people within countries to build these clinical trial networks. Technology increased again because of non-invasive ventilation being formed or becoming a part of practice. And then you had a corresponding increase in quality of life. And moving forward to 2006, this is when we discovered TDP43 as the next sort of big puzzle piece. Technology had increased because we finished the Human Genome Project, and that allowed us to see so much more in biology. I apologize. So, but moving forward to 2012, I would say again, by 2006, the understanding hadn't massively advanced. But by 2012, we now had these new tools to work with. The understanding had increased in the disease. Clinical trials had come further because we started to get multinational clinical trials. We had pharmaceutical industry was starting to be interested in ALS, and technology had started to increase because we could do even more in this genetic discovery. Whole genome sequencing, what's called exome sequencing, these all started to become possibilities. And collaboration further increased because they started to see the breadth of this disease and people coming from other fields into ALS. And finally, quality of life, because we also started to see more assistive technology for people with ALS to be able to communicate better, et cetera. And finally, we moved to 2018. And this is where I think the major changes have really happened in the past 6 years. Visibility, the Ice Bucket Challenge, has finally added to that piece of the pie. Understanding, as I've said, has massively gone up in terms of where we've gone in ALS in the past 6 years. Clinical trials have massively changed in terms of the way that they're going to be approached moving forward in the future. Quality of life has increased because we're getting closer to brain-computer interface. I think there's still a long way to go, and perhaps that one's a little thicker than it maybe should be. But I think the idea that we need to engage with people who are living with the disease is a huge piece in creating this world without ALS. Collaboration has gone to multinational, international collaborative platforms, and I think it's gone way over the roof in the last 6 years, and technology has just massively advanced to the types of things we can do. And so if I bring that together, and I think that they all sort of interact, this is a very non-scientific way to look at this, but if you say that those two lines, the added thickness of those two lines, when they all work together as a foundation, for example, here would be 1993, and I give you one final diagram to sort of show you where my mind is at in terms of the hope factor, I would say for creating that world with ALS, which is somewhere way up there, when we look at 2012 versus 2018, I think all of those things are coming together, and they're really creating an environment of hope that is so much farther than we were 5 or 6 years ago, even though it doesn't necessarily always feel that way. From myself, internally in my brain, it really does feel that way on a daily basis, which is why I talk so long. So I'm going to get in trouble for this later, but in the end, somewhere up there is that world without ALS, and I think we can get there, but thinking about that trajectory, that's where I wanted to leave you on, was that piece of hope. So if you didn't get anything else from that, I hope that this at least shows you that in my brain, I feel like all those things together have led us to this place where we're on an amazing trajectory to be able to get to that world without ALS. And Chicago, Glasgow, I hope you have a great rest of the week, and that's all I really have for you. I know there's a lot. Thank you.